Assalamu alaikum, dear students. It is nice to see you again. Before starting your new lesson, let's have a short review at your previous lessons. If you remember, we studied about the cell theory, about the microscopy, and also we studied about different types of cells and cell organs. In this video, you will be able to understand cell and surroundings of the cell. First, we will study about the membrane and also about the permeabilities. And second, we will study about the basic components of the cell membrane, and we studied about how substances enter and leave the cell. Let's go and talk about it in more details. Before starting the lesson in detailed information, we should first define the cell membrane or membrane itself. In simple words, we can define a membrane as any delicate sheet that separates one region from another, blocking or permitting, means selectively or completely, the passage of substances. As you see this picture, you can find different structures. So the different structures possess different kind of functions. Let's talk about it in more details. So, one of the most important properties of the membrane is their permeability. According to their permeability, we can define or divide the cell into these categories. First, most of the membranes are impermeable. Second, most of them are permeable. And some of the membranes are semi-permeable, means they're specified to admit specific materials. It means they are selectively permeable. As you see in this picture, there are two layers. It means there are two membranes and that face one type of substance. As you see this diagram, it is going to pass from the membranes. So this is called permeable membranes. It means it admits the substances and materials to pass through it. This one, as you see, it is the same substance, but when it faces the membrane itself, the membrane do not admit it. It means when the material and substances face the membrane that do not admit them to enter the cell or get outside of the cell, so that is called impermeable membranes. Permeable membranes are those membranes that admits membranes that could pass inside the cell and could get outside the cell. Semi-permeable membranes are those membranes that they are specified for important ions and molecules. It means they admit some molecules to get inside and admit some molecules and ions to get outside of the cell, but not more molecules or ions. They are specified for special molecules and ions. Let's go forward. The second objective of this video is to understand the components of the cell membrane. So if you know, the cell membrane contains different types of molecules, and mainly there are three types of lipids and two types of proteins. So these different type of structures possess different type of functions. As you know, the phospholipids, they are composed of mainly two regions. One, a polar region, the other a non-polar region. When we say polar and non-polar, you understand that one of the region of the phospholipid bilayer, which forms the first structure of the membrane, is hydrophilic and hydrophobic. For example, one side of the phospholipid layer, the white ones, they are a group of phosphates which are acting as a polar. It means they are attached with fluids outside of the cell and inside of the cell. Both phospholipids are faced to exterior parts of the membrane, and also the non-polar parts, which are the tail of the phospholipid layer, are phased into interior part of the cell membrane. Beside the phospholipid bilayer, we possess different type of another materials like the proteins and carbohydrates, and these are the carbohydrates which combined with the protein and also with the lipid that produce glycolipid, also glycoproteins. So in order to understand their functions, we should know that they're really important, each of them. So in another simple words, we categorize the basic components of the cell into phospholipid, cholesterol, and glycolipid. There are three main parts of lipids that you can find within the cell membrane. Also, we can find two kinds of proteins, protein and glycoprotein. They were specialized for important functions. For example, the phospholipid, which is acting like a barrier. It means it separates the inside of the cell from outside of the cell. It means it do not admit most of the cell substances to get outside of the cell and most of water substances to get inside the cell. Therefore, it acts like a barrier. Another important function that is carried out by phospholipids are this, that they act like signaling molecules, and also they act like enzyme activities. It means they can flood inside the cell membrane to activate different type of enzymes. So the second thing that is going to be thought about is the cholesterol. These small structures that you can see are in green color. They are called cholesterols. Cholesterols are really important for a cell membrane because of their important functions. The most important function that is carried out by cholesterol is the stability of the cell membrane structure and maintaining the fluidity of the cell membrane. It means when the cell membrane lacks cholesterol, the cell membrane quickly breaks down and bursts open. So if we want to talk about the glycoproteins, first we should know what is glycolipid and also what is glycoprotein. Mainly glycolipid and glycoproteins are a combination of the carbohydrate chains that is attached with the lipids and also attached with the proteins. Mainly they are projecting like antennae, and they are involved in important functions. For example, they act like a receptor molecules that could be divided into three categories of signaling molecules. Also, a group of carbohydrates are involved in the cytosis process, that different type of materials are engulfed inside the cell by recognizing of these structures. Also, the third signaling molecule could be thought as a cell adhesion. It means it is the responsibility of these carbohydrate chains that make adhesion between the cells and tissues of the organism. So also they act like cell markers. It means these are acting like identification cards that are specialized for important molecules and ions. If you want to talk further about the next important material that you can find inside the cell membrane, exactly, that is protein and glycoprotein. Mainly, as I thought about the glycolipid, the same thoughts are similar on accepts for the glycoproteins, but the protein itself could be thought in different ways. It means they act quite differently from phospholipid bilayers and also from cholesterol and glycolipids. The proteins could be thought to be like channels that they act like a receptor or signalers, also act as an enzyme, and also it can act as a cell markers. So these were the basic components of the cell membrane and their functions. So now we received after the objectives of our today's video, which is known as how substances enter and leave the cell. Mainly in material exchange between the cells, we can define different type of functions into 5 basic type of cell transport. It means the cell is communicating between each other through these ways. First, diffusion. Second, facilitated diffusion, osmosis, active transport, and also bulk transport. Let's talk about it in more details. Dear students, diffusion. First, let's define it. Simply, we can say that it is a passive process. It means it do not require any kind of energy, during which substances move from region of higher concentration to a region of lower concentration. It means where the molecules and ions are more, it diffuses through kinetic energy or motion law to a region of lower concentration, or where you can find molecules in small quantities. So the diffusion is affected by different factors. So one of the first factors that affects the rate of diffusion is the steepness of the concentration. For example, in this picture, you can see that a droplet of the ink is put into a flask of water, then it is going to be diffused through the areas of this flask, and finally you can see that the molecules of the ink is spread out to all this glass. Another thing is the temperature. It means temperature also affects the rate of diffusion. As you know, in kinetic law or law of motion, temperature affects the molecules motion. It means temperature motivates the molecule to move faster, so the diffusion occurs faster in those solutions that they are hot. Another important thing or factor that affects the diffusion is the surface itself. It means the surface area where solutions is going to be carried out. So when the area of the diffusion is a little big, so the molecules that are going to be spread out, it diffuses faster. Another important thing that affects the rate of diffusion is nature of molecule and ions. In motion law, we understand that when the molecule size are small, they can diffuse or move faster than those molecules and substances that they possess larger molecular sizes. So because of the larger molecular size, more energy is consumed, so molecules and ions which possess larger sizes, they tend to move slowly. Another way that cell communicate with each other is facilitated diffusion. There is no differences between the diffusion and facilitated diffusion, but except of this point that facilitated diffusion means that it is made possible or it is made easy, and this function is carried out by different type of proteins that are embedded inside the cell membrane. For example, the diffusion of substances through transport proteins. This could be the definition of the facilitated diffusion. It means the diffusion carried out by different type of proteins that are embedded inside the cell membrane. So it is provided by hydrophilic areas that allow the molecule of ions to pass. It means here the different portions of the proteins that they are not specific and specific, and every kind of the substances which are necessary for metabolic activities of the cell, they could be diffused by facilitated diffusion from outside of the cell into inside the cell by these protein channel and protein carriers. Also about those materials that they are really short in molecular size, they can diffuse easy. Another important type of communication between the cell is osmosis. It means when the cells communicate with each other to exchange materials which are necessary for metabolic activities or life of the cell. and so they can use different type of methods. One of them is osmosis. Osmosis and diffusion do not have any differences except of a delicate sheet or a membrane that could be defined like this. It means the definition of osmosis is this: water will flow from a region with a lower solute concentration, it means hypotonic, to a region with a higher solute concentration, it means hypertonic. According to this picture, if you put the red blood cell and 3 different type of osmotic pressure that is possessed by solutions, it means we can divide the solutions according to their osmotic pressure into hypotonic solutions, isotonic solutions, and also hypertonic solutions. So in these different type of osmotic pressures, the cells will act quite differently. As you see in hypotonic solutions, the entrance and diffusion of the water is in one way. It means outside of the water gets inside the red blood cell. So it causes to burst and open the cell, red blood cell. The isotonic solution is a kind of solution in which the entrance and exit of the water is equalized. It means it is in a quite reliable quantities. It means the life of the red blood cell could be not damaged. Also in hypertonic solution, you can see that there is also one way of transporting. It means a solution causes the water of the cell to be exit of the cell. So hypertonic solutions causes the cell to be shrinked. The same example is another illustration. In this video you can see that the 3 different bottles that possess 3 different type of solutions in hypertonic solution, hypotonic solution, and isotonic solution, and the effects of these solutions on the plant cells. So in isotonic solutions, you can see that when cell absorbs water, they absorb the water in amount of the need. So the cell is flaccid cell. Now in hypotonic solution, you can see that water is getting outside of the cell, so the cell shrinks. It is also called plasmolysis. It means water is going to be loosed by the plant cell. In a hypertonic solution, you see that the cell is turgid cell. It means it absorbs more water and it stands quite happily. Another important type of exchanging material is active transport. When we say active transport, we should know that active transports are those kind of exchanging material method in which energy is consumed by the cell in order to carry out this function. So in definition of the active transport, we can say that the movement of molecules and ions through transport proteins across a cell membrane against the concentration gradient using energy of adenosine triphosphate or ATP. Simply it means that molecules and ions are going to be exchanged into 2 different types of the regions. For example, the ions where their concentration is low, they should move into region where the concentration is high. In order to carry out this function, the energy is needed, and this energy is provided by ATP. As you see this picture, the function of active transport could be illustrated in these pictures from A up to F. In this picture, you can see different type of structures. First, these regions are called outside of the cell. This is the inside the cell, and this is the phospholipid bilayer, and this is the specified protein channel. So in order to carry out the active transport, you can see that 3 ions of the sodium are attached to their specific region and protein channel. Also, this specific shape is for potassium ions, which is necessary for metabolic activities of the cell. So this place, this is called the receptor, where the adenosine triphosphate attaches and provides the energy that causes the protein to change itself or change its shape to exchange the sodium ions with the potassium ions. Here you can see that energy is provided by ATP or adenosine triphosphate, and adenosine triphosphate will exchange into adenosine diphosphate, and the protein, after receiving the energy, it exchanges its shape. Then 3 ions of the sodium will diffuse from inside of the cell into outside of the cell, and by exiting of the sodium molecules, 2 potassium molecules will enter the cell, and this is the continuation of the active transport. Why it is important? In animals, we can say that active transport is used by nephrons that are located inside the kidney. Why? Because most of the ions and molecules are important in order to observe by the neurons. Also, this process is used by plants for loading the sugar from cells that are going to be produced inside the chloroplasts. It means the sugar are going to be produced by photosynthesis inside the chloroplasts. Also, the plant needs, in order to obtain or reabsorb inorganic ions from the soil, by hair roots. So active transport is used by kidney nephrons in order to reabsorb different type of important molecules and ions. Also, it is used by plants to absorb or load sugar from photosynthesizing cells, also from soil, the inorganic ions. And if we want to talk about the bulk transport, we can say that most of the time cells need different type of substances in order to carry out metabolic reactivities. So first we should categorize the bulk transport that they can be taught as eating, drinking, or engulfing materials or removing materials. But first, these larger molecules or macromolecules that are important for metabolic reactivities of the cell, the cell needed to be observed from inside of the cell or from outside of the cell. So, sorry, once again. Another important method of material exchange between the cell or bulk transport. Why do we say it bulk transport? Because most of the times in metabolic actions, cell is in need of macromolecules. So there should be a different method to observe and receive these macromolecules like proteins or polysaccharides. So the bulk transport is defined like this, that the movement of macromolecules such as proteins or polysaccharides into or out of the cell is called bulk transport. We can categorize the bulk transport into 2 types of transports. One, endocytosis transport, and second, exocytosis. Simply, endocytosis means engulf of materials. An engulf of materials could be categorized into phagocytosis, that means cell eating or uptake of solid materials, and also panocytosis, which means cell drinking, that is the uptake of liquid materials. Also, when the cell metabolic activities is carried out, then it is necessary to get rid of the waste products and also to exchange the materials with another cells that are in need of these macromolecules. So the process in which different type of materials are going to be exit of the cell, that is called exocytosis, or remove of materials. In this picture you can see that the cell membrane is engulfing the food or solid material, so this is called phagocytosis. The second is called perocytosis. As you see, these are small droplets of the liquid which are going to be engulfed by the cell membrane. These projections that you see, these are called receptors, and that they are specified for specific molecules or materials. In other words, when the cell is in need how to receive and how to recognize those important macromolecules, so these are the receptors which act to recognize those macromolecules for the cell interior parts. So after using the macromolecules, again, this process is carried out into reverse action, and these macromolecules, it doesn't matter that it could be a solid or it could be a liquid, it is going to be expelled right off by the cell. For example, different type of bacteria and viruses will act on effect on the cell, and these viruses and bacteria or unwanted materials are engulfed by the cell. But inside the lysosome, that is a garbage collector, you can find many different types of digestive enzymes that will break down those unwanted materials or macromolecules. Then it will expel it outside of the cell by these small vesicles that you see. So dear students, now we reach at the final slide. So this was your today's lesson. See you on next video. If you possess any kind of question, please inform us. Thank you so much.